Okay, once the DNA's been released, once those cells have kind of cooked for 30 minutes and we've got our genomic DNA solution, we move on to the next step, which is setting up some controls plus the actual transformation process. We take our plate of heart infusion agar that has no cells growing on it and working on not the lid side, but the agar containing side of the plate. Note that, please. We will divide the plate into quadrants using our Sharpies, voila. And then we will label the quadrants according to the directions in the lab manual. One will contain SDRR live cells, which will be transferred from the plate that I showed you in the last video. One will contain SDRS cells, again, from the plate I showed you in the last video. One will have DNA alone. That's the solution that's currently, or we just removed from the water bath. There shouldn't be living cells in there. It should just be lysed cells plus their genomic DNA content. And the other one is where kind of the magic of the transformation should happen. That should be labeled STRS plus DNA. That's where we will be mixing together live susceptible cells with that genomic DNA solution from the resistant strain. So once we've labeled along the edges, again, on the agar side, not the lid side, right? You should have gotten by now that that's important, right? Then we're gonna go ahead and strike our Bunsen burner because we need to keep everything sterile here. So I'm gonna light the Bunsen burner. I'm gonna take my loop, and the first one I'm gonna do is the DNA solution itself. So I'm gonna flame the loop, as I showed you in the previous video. Once it's red hot, I know it's sterile, I'm gonna open the tube, cool the loop on the inside side of the tube. It'll melt the plastic just a little, that's okay. Take a loop, it's almost like when you were a kid and you'd be blowing bubbles, so take a loop of the solution and just make a little line down the middle of the DNA quadrant. Leave that on the bench in front of you. Flame. And then we're gonna go back into that tube. We're gonna get another loop full of DNA solution, cooling it on the side. You can see that I've got some solution there, and I'm gonna just put a drop of it in the middle of the quadrant that is labeled STRR plus DNA. So right in the middle, I'm just dabbing it on there, right? So just in the middle. I know it's a little hard to see. Flame. And then I'm gonna go into the STRS plate. I've got cells, it's STRS. Cool the loop on the edge by poking it on the edge of the agar plate. You'll hear a little sizzle. Gather up some cells just by touching it and pulling the loop back. And I can see a white film of cells on the end of the loop. And then I'm gonna go into that little bit of DNA that was sitting in that relevant area of the quadrant and make a little sort of soup. Gonna like mix them together, okay? So I'm mixing it together in the little area in the quadrant. Got a little soup of DNA solution plus STRS cells. A little goes a long way. Flame the loop. And then I'm gonna go back into the STRS cells, cool the loop, gather up cells, and then I'm gonna go into the quadrant labeled STRS, and I'm just going to drag the loop down, okay? Just making a little line down the center. I've inoculated that quadrant now. That's what this is called, inoculating, or transferring. Flame the loop to sterilize. Go into the STRR stock cells, right? Cells growing on that plate. Cool the loop. We'll sizzle on the outside. Grab a few cells. go into the STRR labeled quadrant. Okay, make sure not to screw that up. And then draw a little bit of line down. I'm inoculating that quadrant with the loop. Now I'm going to put the lid on the plate, the 4 quadrants, flame the loop. I'm going to have to flame it for a bit just to make sure I got all the cells that were left on it removed. They're going to burn up. You can hear them screaming as I toast the loop. All right, and then I'll lay my loop down to cool. I'm going to turn the gas off. And now what I have is I have a plate with 4 quadrants, and on the plate there's a quadrant that has STRS cells. That's just a transfer. They should grow because there's no streptomycin in this HI media. It's just media, just food for the bacteria. I also have a quadrant with STRR cells. They should also grow just fine on this media. There's no selective advantage for them relative to the susceptible ones because at this time there's no streptomycin in the media. Then I have DNA alone. That really shouldn't grow because the treatment with the detergent and the heat should have killed everything there. So that's a bit of a control. And then I have the quadrant where something interesting should be happening. The STRS strain is live cells, and they're sort of sitting there on the plate surrounded by genomic DNA from the resistant strains. That genomic DNA should be taken up by the susceptible cells, which are naturally competent, meaning they can be transformed. They take that DNA up and they'll incorporate some of it into their genome. Some of them will incorporate the AAD2 gene in, which will effectively transform those cells into streptomycin resistant cells. When we're done with this, we incubate it at room temperature basically until the next lab period, although they'd be ready to look at the next day. We just don't have that on our schedule. So this will be like 1 week later that we'll look at them. What we are expecting to see is growth on all the quadrants except for the DNA only quadrant, which is a control for cell killing. And the incubator is just basically like a sealed, kind of box that's sitting there at room temperature, kind of like an unplugged fridge. And that's the basic setup for the first stage of this lab.